In today's video, we're going to be taking a look at genetic engineering for A-level biology. If you see a certain point on this page that you want to skip to, take a look in the video description below, as I've listed all of the respective timestamps. Otherwise, let's go ahead and get started. The first thing to understand is that genetic engineering is the branch of biology in which techniques have been derived to remove or alter genes, to synthesize new genes, and to also transfer genes from one organism to another. So recombinant DNA is when we get the DNA from two different sources and we combine it to make a new piece of DNA, which is called novel DNA or recombinant DNA. Now, when we go ahead and we put that recombinant DNA into another organism, that organism is called a recombinant host, right? Because it has that recombinant DNA within it. Now, another way for saying recombinant host, another word, is a genetically modified organism, okay? So in organisms that have recombinant DNA within them are called genetically modified organisms. So why do genetic engineering in the first place? Well, the reason why is because by doing genetic engineering, we can obtain pure proteins, okay? And so by doing this, we can then obtain a desired trait within an organism, which could be very useful in, let's say, crops, okay? Which we'll get to later on, maybe in cattle, right? In dairy farming and milk production. So genetic engineering is important because it enables us to have a target gene, a target protein being produced within an organism. Now, the steps of genetic engineering, we'll quickly go over the steps of genetic engineering, and then we will actually take a look at the processes occurring at each step. So the first thing that we have to do is we have to obtain a gene, and there are three methods. The first one is that we could use reverse transcriptase. The second one is that we could chemically synthesize the gene. Okay, so if we have a gene and we know the sequence of amino acids that make up that gene, and using genetic data, we can identify which three nucleotides make up each of those individual amino acids, then what we can go ahead and do is we can make those amino acids. So we can compile the three respective nucleotides together to make the first amino acid, then three other nucleotides to make the second amino acid, and then so on and so forth until we have the full gene. The third and final method that we could use is we could use restriction endonucleases to basically just cut out a specific gene from an organism, right? Cut out a specific target gene from an organism. What we then do is we do cloning. So we make heaps and heaps of copies of that gene using DNA polymerase, right? And for example, we could use PCR, which is polymerase chain reaction. What we then go ahead and do is we do transformation. So this is when we put that gene, that target gene, into an organism, okay? So into a host. And we can do this with the use of vectors. What we then go ahead and do is we do identification. So we can use gene markers, right? We'll take a look at gene markers a little bit later on. So that is basically just to check to see if the target gene has actually been uptaken by the host cell. The host cells that have uptaken the gene will then be made in large-scale production. So we will do large-scale production of the host cells that have successfully uptaken that target gene that we put in. So now let's take a look at reverse transcriptase. As we saw in the transcription versus translation video, in transcription what occurs is we have our DNA double helix molecule, it's unwound, and then with RNA polymerase we synthesize an mRNA strand which goes on to translation. Now, as we discussed, a method of obtaining a gene is using mRNA to obtain a strand of complementary DNA using reverse transcriptase. And so the way that this actually occurs is we get the mRNA strand and then we use reverse transcriptase to get a strand of DNA with complementary nucleotides to that mRNA strand, and that is called complementary DNA. Now, DNA polymerase, what it's going to do is to a single strand of DNA, it will add the complementary nucleotides and it will create phosphodiester bonds between adjacent nucleotides, creating a fragment, okay, a DNA fragment, right? So we're going to have a phosphate sugar backbone that has been formed. Now, DNA ligase, what that's going to do is when we have two separate DNA fragments, so there is a space between the phosphate sugar backbones of those DNA fragments, DNA ligase will come and it will create the phosphodiester bonds between them, creating one whole fragment. Now, what I want to clarify really quickly is that when we have the mRNA strand and we get a DNA strand using reverse transcriptase, that DNA strand is said to be complementary to the mRNA. So it's termed as complementary DNA. It's important to understand that this isn't the only complementary DNA that occurs, right? If we have DNA polymerase working to create another DNA strand, to create a double helix DNA molecule, that strand that's being synthesized will be complementary to this DNA strand. So it might be a little bit confusing, but it's important to understand. We can have mRNA, and then we can have a complementary DNA strand to that mRNA. And then we get another DNA strand that is complementary to the first DNA strand, which is complementary to the mRNA. So it's important to understand that complementary DNA, there's not just one complementary DNA strand occurring. There are more. It just refers to the complementary nucleotides that are occurring. So now let's move on to restriction endonucleases. So restriction endonucleases are enzymes that cleave or cut a DNA double strand at a specific sequence of bases called a recognition sequence. Now, the way that restriction endonuclease enzymes can cut is that they can either cut directly through the DNA double strand, which would then leave these blunt ends. Okay, so these regions here are called blunt ends, or the restriction endonuclease enzyme can cut in a staggered fashion, leaving sticky ends, which are bases that are unpaired. Okay, so the sticky ends will be these bases that are unpaired. Now, it's important to see that in these sticky ends, let's say we had A and, let's say, C, actually, A and C, and then on this other sticky end, we will therefore have thymine and guanine, right, because ATCG pair in DNA. These, if we read from left to right each of the bases on each sticky end, they're actually opposites, right? AC and TG, they're opposite nucleotides, they're opposite bases. And so when this occurs, when they are opposites, they are said to be a palindrome. So now that we've taken a look at two of the processes that we can use to obtain a gene, right? Reverse transcriptase to get complementary DNA. And then we've taken a look at restriction endonucleases to cut a specific gene out of a long DNA nucleotide sequence. Before we actually put the gene into an organism, into a host cell, what we can do is we can add promoters to the gene. And so promoters will enable transcription of that subsequent gene. Now, without promoters, we would then have to add the gene close to a promoter that is already present in the organism's DNA nucleotide sequence, which could be difficult, and it may disrupt other gene expression, or it may not actually even express the specific gene that we're inputting. So we add a promoter region to the DNA nucleotide sequence, which will enable RNA polymerase to begin the transcription of that gene, right? Because that's what we want. We want transcription and then translation for the protein synthesis of that target protein. What we can also do is we can add marker genes. So marker genes are basically genes that we will use to make sure, right, as we talked about in identification, we will use them to see if the organism, if the host cell has uptaken the gene that we inputted, right? Because we can put a gene in, but it may not actually be uptaken in the organism's genome. Now, a type of marker gene that we could use is a fluorescent marker, right? The GFP gene from jellyfish fluoresces when placed under blue to UV light, okay? And so if we have the organism, we have the cell that is now a host cell, we've put in a new gene with the fluorescent marker. If it has successfully uptaken the gene, then when we put the organism under blue to UV light, it will fluoresce. And that way we know that the gene has been successfully uptaken by the organism, by the host cell now. So now let's take a look at microarrays and gene profiles. But firstly, what is a gene profile? So in an organism, let's say for example a human, we have over 20,000 different genes. Now some are switched on, so they are being expressed. So mRNA is being synthesized, right? It's being transcribed. The DNA is being transcribed into mRNA. And other genes are switched off, so they aren't being expressed. Now, the gene profile is the pattern of genes that are being expressed at any one point in time within an organism. Now, gene expression profiling is where we look to see the gene profile of an organism using, for example, microarrays. Now, a microarray has thousands of different spots, and within each spot we can put some DNA, which is going to be called a DNA probe. Now, we put in a single strand of DNA that codes for a specific gene or allele, and we record what DNA probe in the microarray has the DNA strand for which specific gene or allele. Now, these DNA strands will be complementary to the DNA strands that we will get from our sample organism. Now, we also have to make many clones of these DNA strands because this is actually what will enable us to identify the degree to which each gene is being expressed within the sample organism. Okay, so we have the DNA probe sorted. Now, we move on to the sample organism, and as we discussed before, genes that are being expressed will have mRNA being synthesized. Genes that are not being expressed, mRNA for them will not be synthesized. So we extract the mRNA from the cells, and we use reverse transcriptase to get complementary DNA strands, as we discussed earlier on in this video. So we then get our complementary DNA strands, and now we have this mixture of complementary DNA strands and mRNA. We label the complementary DNA strands with gene markers, right, with fluorescent gene markers, and then we treat the mixture in order to eliminate the mRNA strands and leave only the complementary DNA strands that are labeled in the mixture. We then wash over all of the complementary DNA strands over the DNA probes, and what happens is the complementary DNA strands will attach, so they will bind to their complementary strand that was in the DNA probe. Now, and then what will happen is hybridization will occur. So when the complementary DNA strand from the sample organism binds to the DNA strand that we placed in the DNA probe, they will hybridize. Then what we do is we use a laser to scan the microarray and the microarray slide. And what will happen is where hybridization has occurred, we will see a color, right? The fluorescent gene marker from labeling the complementary DNA will show. So that will mean that that respective gene is being expressed in the sample organism. In DNA probes where there is no fluorescent marker, where there is no fluorescent color being shown, that means that there was no complementary DNA from the sample organism that attached to it. Okay, so that means that that specific gene is not being expressed in the sample organism. Now, we're going back to the mini clones. So because we had many clones in the DNA probe, so many clones of the same DNA strand for each respective gene or allele in the DNA probe, when we use the laser scan, the more complementary DNA strands that hybridized with their respective DNA strand for the respective gene, so the more that there are, the more intense the fluorescent color will be. Okay, so we have many clones of that same DNA strand. So let's say this here is one gene. Okay, let's say this here was one gene, and we had the DNA strands for that one gene, and we had many of them, many clones. The complementary DNA will come and bind. If there are many complementary DNA strands binding and hybridizing, then there will be a much stronger fluorescent color than if there are only a few complementary DNA strands binding. And so that will give us an indication of the degree to which that specific gene is being expressed in the sample organism. So now let's take a look at microarrays in genome analysis. So if we want to compare the genomes of two different organisms that are closely related, let's say, what we can do is we can extract the DNA and then separate the DNA strands so that we get single-stranded DNA, as we do in polymerase chain reactions. And then we can splash that DNA over the microarray, which contains DNA probes containing single-stranded pieces of DNA for a specific gene or allele. And then we can use the laser to scan the microarray and identify which genes are being expressed in each of those respective organisms. Maybe one organism has more genes, maybe it has less, and that could be due to a mutation, okay? And so using this information, we can identify differences between different organisms. Now, biological databases are stores of biological information that can be used by scientists for research and development. Now, there are a couple of different types of databases. We can have nucleic acid and genome databases, so where we sequence DNA and obtain important information. And we can also have protein sequence databases, where we take a look at which amino acid sequences make up different proteins. So now let's take a look at vectors. In biology, a vector is a tool that can be used for the transfer of genetic information into an organism. Now, plasmids are generally used for the transfer of genetic information into bacterial cells, so into bacteria, whereas viral vectors, so viruses, are generally used for the transfer of genetic information into eukaryotic cells, like in humans, for example. Now, the way that a plasmid can act as a vector, so what we will do is we will have the genes, so we will have a target gene within an organism, and we will use a specific restriction endonuclease to cut that specific gene out of the organism, out of the long DNA nucleotide sequence of one organism. Now we have the specific gene that we want. We go to the plasmid and we use the same restriction endonuclease to cut out a section of DNA that will leave sticky ends that will be complementary to the sticky ends of the desired gene. What will then happen is the desired gene will be incorporated into the plasmid, and then DNA ligase will come along and it will join the DNA fragments together, making a recombinant plasmid. Now, if we want to put this recombinant plasmid into a bacterial cell, right, because we want the specific gene to be expressed, we want a specific protein, maybe for example insulin, to be produced, what we will then do is we will place the plasmid in a medium with bacterial cells and calcium ions. And when we heat this medium up, the plasmids will actually enter through the cell wall of the bacteria and the cell surface membrane, and this will enable the uptake of those recombinant plasmids into the bacterial cell. And then what will probably happen is the respective gene will be produced, right? The protein will be produced. And the way that we can actually test to see if the desired gene has been uptaken is by using a gene marker. So once we've cut the desired gene out of one organism, we can label it with a gene marker so that after it's entered into the bacteria, we can test to see if the bacteria has successfully uptaken the recombinant plasmid. Now, viral vectors, as it states in the name, is the use of viruses. So because viruses are so good at entering and targeting specific cells, we can use that to our advantage to deliver specific genetic information to a specific cell that may be mutated. Now, viral vectors are generally used in human cells, okay? And so when we use viral vectors and we deliver genetic information into a human cell, that is called transduction, okay? So it's going to be a transduced cell if it receives genetic information. So human cells receiving genetic information using a viral vector will become transduced cells. In plasmids, what will happen is if we deliver genetic information to a bacterial cell using a plasmid, it's going to be a transformed cell, right? So the two different types of viral vectors that we need to know are retroviruses for severe combined immunodeficiency, and then the adeno-associated virus for Leber's congenital amaurosis. Okay, so these are the main two viruses that we need to know as viral vectors. Now let's also look at gene guns. So gene guns are very efficient actually to deliver genetic information in plants, although they can be used in other animals as well. So what happens is we have a tungsten pellet coated in DNA, and when it is shot, the tungsten pellet is actually stopped, and then the DNA, that propulsive force, throws out the DNA of the gun into the plant. And it actually enters the plant, and it's actually a very effective way of delivering DNA into the plant cells, into the plant DNA. Now let's take a look at the difference between gene therapy and gene editing. So gene therapy is where we add a normal allele to DNA that has been mutated, and it has a non-functioning allele or a bad allele. Whereas gene editing is where we add or remove or alter DNA at a specific location. Now, gene therapy, so as we discussed a little bit before, we can use viral vectors in humans. And when we use the viral vectors and we deliver genetic information to the human cell, that cell will now become a transduced cell. And in order to do gene therapy, right, to deliver a good allele, we can either use viral vectors, as we've discussed here, or we can use liposomes. So liposomes have a phospholipid bilayer, and they can pass through the phospholipid bilayer of the cell surface membrane to reach the target cell, to reach its nucleus, to deliver the normal allele. Now, viral vectors and liposomes have their advantages and have their disadvantages. Firstly, a liposome is it has a phospholipid bilayer with the normal allele in its center, and the liposome can actually pass through the phospholipid bilayer of the cell surface membrane in order to reach inside the cell and deliver the respective DNA. Now, the advantages of the viral vector in comparison to liposomes is that viral vectors are much more efficient at actually delivering the gene to the target cell. However, the liposomes may be able to carry larger genes than that of the viral vectors. Okay, so viral vectors will only be able to deliver a smaller gene compared to a liposome. However, they will be more effective and more efficient at delivering that gene than the liposome. So that's it for this video. If you found it helpful, take a look at some of the other content on my YouTube channel. Otherwise, thanks very much for watching, and I hope you have a great day. Bye-bye.